Okay, welcome to Ibrahim Academia. Today we are going to discuss the most expensive treatment in the world, or the most expensive medicine of the world. It is not actually a medicine, a pill, a syrup. It is a gene therapy. The most expensive gene therapy in the entire world is Lenmeldy. It is also called gene autotemcel. This gene therapy is proposed for a type of disease which is called MLD, Metachromatic Leukodystrophy. In MLD, there is a missing protein, a missing gene or a dysfunctional gene which is called ARSA. ARSA gene, aryl sulfatase A enzyme. This enzyme, this gene, it occurs on chromosome number 22. This chromosome number 22 protein or gene is dysfunctional. It is mutated and it is autosomal recessive disorder. The child is born with loss of motor coordination, with seizures, with paralysis, no movement of the feet, no movement of the hands, and also speech problem or the cognitive problems. All these problems are occurring in MLD child, and it is completely fatal disease. Within 5 years or within 10 years after the onset of the symptoms, the child moves towards death. MLD child, the brain of the MLD child, from the brain I have taken just one cell, the neuron of that MLD patient. This is a healthy neuron, but in case of MLD, the loss of the myelin sheets happens. So that's why the neuron functionality disturbs. Lenmeldy gene therapy is also called gene autotemcel. For what kind of disease this gene therapy has been adopted in March 2024 and how much it costs? It costs around 4.2 million US dollars. This much. MLD, Metachromatic Leukodystrophy, the white matter of the brain which is composed of myelin sheets. And who composes this myelin sheet? This is composed by a specific type of cells which are called oligodendrocytes. These are the supporting cells, also called glial cells of the brain. These oligodendrocytes have some sort of problem. And what kind of that problem is? The oligodendrocytes are deficient of the ARSA gene, ARSA. This is a type of enzyme which actually metabolizes toxic lipid-like substances called sulfatides. If these sulfatides accumulates inside the oligodendrocytes, then the oligodendrocytes will lose their function. And what is the function of the oligodendrocytes? They form the myelin sheets which surrounds the axons of the neurons. Now the patient with MLD has no myelin sheets around the neurons, and that baby has loss of motor coordination, seizures, and cognitive problems. He or she is deaf, she is blind too. All these problems are occurring because of the ARSA gene deficiency or the missing of the ARSA or the mutated ARSA gene. And eventually that baby is losing his or her life within 5 years. It is very fatal disease, MLD. How to treat this kind of disease? For this, there is Lenmeldy gene therapy. What's inside this gene therapy? Let's go and explore this. You have to correct the ARSA gene inside the squamous cells for the peripheral nervous system and inside the oligodendrocytes for the central nervous system. And how to correct that gene? For that gene, you have to do the gene therapy. Take a lentivirus. I took a lentivirus, and that lentivirus I insert the human ARSA DNA, ARSA gene segment. ARSA gene segment has been inserted and the DNA or the genome of the lentivirus. The ARSA gene segment has been inserted successfully in the genome of the lentivirus. Now you have to take the HSC cells, the hematopoietic stem cells from the MLD patient. Where are the HSC cells? The HSC cells are present in the bone marrow. So take the bone marrow of that patient and took some HSC cells HSC cells has been collected from the patient, those HSC cells are then diffused with the lentivirus. The genetically modified lentiviruses has been reacted with the HSC cells of the patient. Some of the HSC cells will then absorb the ARSA gene segment of the human. These cells will then be called as a Lenmeldy cells. Now these Lenmeldy cells has to be cultured inside the lab to increase the number of these cells. When the number increases, you have to make space inside the patient's bone marrow. For this purpose, you have to do chemotherapy. For chemotherapy, there are certain drugs which can kill the existing diseased HSC cells. After creating some space inside the bone marrow, you have to insert those corrected HSC cells intravenously. Some of HSC cells will establish here. They will make some room and they will reside inside this. Those corrected HSC cells then can form corrected blood cells. The question is, there is a big question inside it. The problem I am going to correct is for the oligodendrocytes or for the squamous cells. Why I am correcting the blood cells? Oligodendrocyte is not a blood cell. Squamous cell is not a blood cell. Then why I am correcting the blood cell? This is a big question. To answer this question, those HSCs, they can form all type of blood cells. And in those blood cells, there is one type which is called monocyte. This is also a blood cell. That monocyte can travel to the brain and that can cross the BBB. Each brain cell is surrounded by a protective sheet which is called blood-brain barrier. That BBB can be crossed by the monocyte, and those monocytes which cross the BBB, they can then act as microglia-like cells. What are microglia-like cells? These are the resident macrophages inside the brain cells. So those monocytes then act as our resident macrophages, and you already know that those monocytes has been corrected with the ARSA gene. The ARSA enzyme can be secreted from the monocytes, from the microglia-like cells, and those secreted ARSA can be taken up by the oligodendrocytes near it, because the oligodendrocytes has a receptor for the ARSA protein which is called M6P, mannose 6-phosphate receptor. That mannose 6-phosphate receptor can take the ARSA protein that can be endocytosed inside the oligodendrocyte. Now the oligodendrocyte has a prepared ARSA protein from the outside. This process is called cross-correction. So Lenmeldy gene therapy is called cross-correction method. The oligodendrocytes now has ARSA protein, and that ARSA protein can now break down the sulfatides inside the oligodendrocytes, and when much of the sulfatides has been broken down by the ARSA, that oligodendrocytes can now form myelin sheets which can protect the neurons, and the baby can get rid of the disease. This is a very much complex process, and it is a personalized therapy. Only one patient can be treated with this kind of treatment.